Hello, SS2. You're welcome again to our online class. Okay, and this is lesson 2 of reproductive systems in vertebrates. The first time we looked at the structures of the male and female human reproductive systems, wherein we saw the parts and functions. Okay, and then what else did we do? Yeah, we looked extensively at their structures, the pictures, diagrams of the male and female reproductive systems of man. So to continue further in lesson 2 today, we'll be looking at the structure of the mammalian gametes. Now, what do we mean by gametes? Sex cells, that's correct. So we have the male sex cell, the female sex cell of human beings. Now the male sex cell of human beings is called the sperm. Okay, the sperm cell, plural, sperms, or you can also call it spermatozoon, plural, spermatozoa. Okay, while the female gametes of human beings will be called the egg or ovum. You can also call it eggs or ova for plural. Now, the reproductive sex cells are known as the gametes. Okay, so now you know what gametes are. They are just the sex cells, the ova, the sperms. Now, the formation of gametes or gametogenesis takes place in the gonads. The formation of gametes is called gametogenesis. So the formation of the sperm cells, egg cells are called gametogenesis. Okay, so and this gametogenesis takes place in the gonads, which are the sex organs. Okay, the gonad of the human male will be testis, the testicles. Okay, while that of the female will be the ovaries. We have two, so ovaries. Okay, so taking a look further on the human female and the male sex cells, let's take a view of what these sex cells look like. So taking a look at the human egg, you see we have different components: the cytoplasm, the corona radiata, which are also called the follicular cells. We have the zona pellucida, also called the jelly coats. We have the cortical granule. We have the nucleus. So these are the components of the human egg. And then for the human sperm, we have 3 major parts. We have the head, the midpiece, and the flagellum or the tail. Now the head comprises the acrosome, the nucleus, and the centriole. The midpiece comprises the mitochondria, while the tail comprises the flagellum. Now, the axoneme are actually structures found within the flagellum that will help the motility of the flagellum. Okay, it will aid the motility of the flagellum. Now, so these pictures are basically the human egg and the human sperm, what they look like. Okay, so that's the structure. You can see that the shape of the human egg is oval, is round. Okay, it's oval shaped, while that of the sperm cell, it's cylindrical, running long. All right, so that's the appearance if you view them under the powerful light microscope or the electron microscope, being that they are both microscopic. Now, taking a look at the human ovum, we want to see the functions of the parts. Now, taking a look at the zona pellucida, which is also called the jelly coat, what is the function So you see that that roundish membrane, okay, the covering of the egg, okay, is the zona pellucida, and it's also called the jelly coat. And what it does is that it contains okay, it helps prevent polyspermy. Okay, aside that, it contains glycoproteins, which is the combination of carbohydrate and proteins, okay, that protect the inner content of the ovum until the acrosome reaction of fertilization. So it just helps to prevent polyspermy. That's the function of the zona pellucida, as well as forming a containment of the inner structure content of the ovum. And then there you see the nucleus of the follicular cell. Okay, we have so many nuclei of the follicular cells, so many of them, right? And then you see the corona radiata, the follicular cells, so many of them as well. What does the corona radiata do? It protects, okay, they each will protect the inner layers of the ovum, and they will also prevent polyspermy. Now, what about the cortical granule? It will also prevent polyspermy. But what it does, okay, how it does this is that it releases enzymes. So these cortical granules that you can see, many of them inside the cytoplasm, and many of them, right? Okay, they will release enzymes during fertilization, and these enzymes will help harden the zona pellucida. Okay, and this hardening will prevent polyspermy in the sense that no more sperms can fertilize the fertilized egg. Now, when we talk about polyspermy, it's all about the tendency of the egg cell to be fertilized by more than one sperm. Polyspermy is the tendency of an egg cell to be fertilized by more than one sperm. Now, just a sperm is supposed to fertilize the egg cell to produce a normal human of 46 chromosome number. But whereby multiple sperms do this fertilization of the egg cell, it can be lethal and can affect embryo development, and can even lead to the death of the embryo. So that's why the cortical granules, they will release enzymes that will harden the zona pellucida after the egg has been fertilized by one sperm. So this hardening will prevent the further penetration of other sperms that may have come in later, or they may have come around the egg later, so they cannot permeate or penetrate this fertilized egg anymore. And this will help prevent, of course, polyspermy. Now, the mitochondrion, plural mitochondria, what it does is that it, is the powerhouse of any cell, okay? And cellular respiration occurs here. So ATP will be produced, okay? Energy in the form of ATP will be produced, and this mitochondrion also contains mitochondrial DNA, which is different from the nuclear DNA. The mitochondrial DNA is only contained in the ovum, all right? And then it will be passed on to the offspring. So the mitochondrial DNA is passed on from the ovum to the offspring. The nuclear DNA is different from the mitochondrial DNA. The nuclear DNA is coming from both male and female, right? But the mitochondrial DNA is coming only from the female gamete to the offspring. Okay, so the egg cell here also contains the yolk in the cytoplasm, which will provide food at the early stage of development of the embryo for the embryo. Okay, so the embryo will feed on the yolk until implantation, where placenta will be formed. But that initial stage of embryo development, the embryo is feeding on the yolk, which is contained in the cytoplasm. Now, the cytoplasm is forms a containment for organelles of this egg cell, such as the nucleus, mitochondria, cortical granules, all right? So they all will lie in the cytoplasm. And then take note that the nucleus is haploid, okay, 23 numbered chromosome, so that when it fuses with the haploid number nucleus or haploid number chromosomes found in the nucleus of the male of the sperm, it will give you a viable zygote that can develop into a normal human being. Now, taking a look at the structure of the male sperm cell, okay, spermatozoon, Z-O-O-N, zoa, Z-O-A, that's the plural. So spermatozoa is the plural form of spermatozoon. So this is actually one, so it's spermatozoon, Z-O-O-N, should be the suffix or the latter part. So you see that like we said, the spermatozoon or the sperm cell, okay, is having three major regions of the head, the midpiece, and the tail. Now the head, all right, comprises the acrosome at the tip, okay, and this acrosome is a lysosome, okay. It contains lytic enzymes, okay, that will help break or digest the zona pellucida and will help that the content of the nucleus of the sperm fuses with that of the nucleus of the ovum, leading to fertilization and the development of a zygote. So so take note that the acrosome found at the tip or the end of the head of the sperm, okay, contains special enzyme, lytic enzymes, that will help break the zona pellucida or the jelly coat, leading to the permeation or entry of the sperm into the sperm nuclear content into the ovum. Now fertilization, you know, is the fusion of the nuclear content of the male sperm and the female egg, right? Okay, so the midpiece, of course, contains mitochondria, and of course it's the powerhouse of any cell. Mitochondria, plural, mitochondrion, singular, so there are lots of them. So mitochondria, they are the powerhouse of any cell, and they will provide energy in the form of ATP that will help the sperms, okay, it will help that the flagellum, okay, the flagellum of the sperm can move. So the mitochondrion provides energy that will aid the motility of the flagellum of the sperm cell. So the sperm cell swims, okay, the flagella is for movement, it swims, okay, and this swimming, of course, needs energy. So mitochondrion will provide energy in the form of ATP that will help the propulsion of this tail or flagellum of the egg cell, sorry, of the sperm cell, okay, of the sperm cell. So there you go. So comparing the male gamete or the spermatozoon with the female gamete or the ovum, okay, let's take a look at the male gametes. Now the male gametes, okay, there are many. They are called sperm cells, okay, or spermatozoa, okay, and they are produced in the testes by a process called spermatogenesis. Now the gamete is unicellular in nature, that means it is single-celled. Now the sperm consists of a head which contains the nucleus, the acrosome, okay. It also has the middle piece and the whip-like tail called the flagellum. Whip-like, okay, because when it's swimming, it's actually going like a whip, okay, moving, you know, like a whip. So it's whip-like, okay, like it's swimming, okay. Okay, so you know the way the flagellum moves, right? Like whip-like movement. So the human sperm is about 0.05 millimeter long, so it's quite too little and microscopic, so it cannot be seen with the naked eyes because it's just 0.05 millimeter. That's very tiny or minute. So you'll be needing a powerful light microscope or an electron microscope to view these sperms, and even the way they move. You'll be needing an electron microscope to do that. And the sperm cell is smaller than the egg cell. Now taking a look at the female egg. Okay, the female sex cells or the gametes are called the eggs, singular egg, or ova, singular ovum, and are produced in the ovaries by a process called oogenesis. So oogenesis is the biological term for the formation of egg cells in the ovaries of the female animal, while spermatogenesis is the biological term for the formation or production of the sperm cells in the testes of the male animal. And the ova are larger than the sperms. Okay, and each ovum is about 0.1 millimeter in diameter. It consists of the cytoplasm, a nucleus in the center, granules, and yolk droplets. Okay, so the cortical granules that will secrete the enzymes, right? Now the yolk provides a source of nourishment for the embryo, especially at the early stages of development. So before implantation, before the placenta is formed, before the fetus starts deriving its nourishment from the mother, okay, that initial time of embryo development, okay, the embryo feeds on the yolk contained in the fertilized egg cell. So taking a look at spermatogenesis and oogenesis. Let's compare. We already know what spermatogenesis and oogenesis mean, right? In spermatogenesis, all right, take note that this occurs in the testes, specifically the seminiferous tubules, and it begins to happen during puberty in males. Now, the spermatogonia, singular spermatogonium, okay, will be the stem cells. Spermatogonia are stem cells. So, picking a spermatogonium, which is just a stem cell, okay, you will see that the spermatogonia or spermatogonium, okay, they are diploid, or having 46 number chromosomes. But you know that we're talking about spermatogenesis, that is sperm production, and you know that the number should be haploid. So take note that this diploid will have to divide to haploid. So how will this happen? So spermatogenesis is just a process whereby the diploid numbered spermatogonia will differentiate to become haploid spermatids. Now what basically happens is that the spermatogonium, which is haploid, okay, will differentiate to become a primary spermatocyte, which is also diploid. Spermatogonium, which is diploid, okay, spermatogonium, take note, is diploid, all right, it will differentiate to become a primary spermatocyte, which is also diploid, and this happens at puberty. And at this puberty, the first meiosis happens in the male animal, which includes male human beings. So the first meiosis will entail that the primary spermatocyte differentiates to become two secondary spermatocytes. And then the secondary meiosis will happen. The secondary spermatocyte will then yield 4 spermatids. That is, each secondary spermatocyte will produce 2 spermatids, giving a total of 4 spermatids. Now these spermatids will then become the immature sperms. Each of them will become one immature sperm. So that is a total of 4 immature sperms. So these immature sperms will be stored in the epididymis until they are mature and can be released or ejaculated to the exterior. However, in oogenesis, you discover that this occurs prior to the birth of the female animal. So oogenesis has already started while the female animal is still in its mother's womb. So what happens is that the oogonia, which are stem cells, okay, and they are also diploid, having 46 numbered chromosomes. But you know, we are talking about ovum production, which is egg cell production, so it should differentiate into haploid number. So oogenesis is just that the oogonia, being diploid, becomes or differentiates to become secondary oocyte or ovum, as the case may be. Now the thing is that oogonia, we said, are stem cells and they are diploid. Now picking one oogonium, singular oogonium, N-I-U-M, if it is singular. Now picking one oogonium, all right, it will then, okay, differentiate into primary oocyte, which is also diploid. This happens prior birth, when the child is still in the mother's womb. Now that means that the female child will be born into the world with all her primary oocytes, all the primary oocytes she will ever need in her lifetime. But there will be a pause until puberty sets in. Now during puberty, the primary oocyte will undergo first meiosis to produce a polar body and a secondary oocyte. Now second meiosis will also occur at puberty to produce a secondary oocyte and 3 polar bodies. And then if fertilization occurs, the secondary oocyte produced will then be fertilized by a mature sperm, which has been released by the epididymis. Okay, to become a fertilized ovum, which will now differentiate into the zygote and will give rise to the embryo, the fetus, and the newborn. Okay, and then the polar bodies will then degenerate because they are not viable and cannot be fertilized. So the secondary oocyte plus a mature sperm cell will give you a fertilized ovum. Will give you an ovum. The ovum is fertilized and it is mature, and it is this one that can now become the zygote. And the ovum is haploid. The secondary oocyte is what? Haploid. The sperm cell is also haploid, right? But when they come together and to form the fertilized ovum, the fertilized ovum will then differentiate to become 2 end zygotes. So you now have a diploid numbered zygote and a new 46 numbered chromosome human being forms. Okay, now that is all about spermatogenesis and oogenesis. So secondary oocyte is not yet called ovum until fertilization takes place. Then you now have the ovum. So the ovum is fertilized secondary oocyte. But if no fertilization takes place, then the secondary oocyte together with the polar bodies will just be flushed out during menstruation. Okay, so take note that take note. All right, so that's just it. And you should know the number of secondary spermatocytes, for example, formed during the first meiosis of spermatogenesis. You know it's 2, but at the second meiosis, 4 spermatids will then be formed, which will become the sperms. And then for the first meiosis of oogenesis, 1 polar body, 1 secondary oocyte, but the second meiosis, 3 polar bodies, 1 secondary oocyte, which if fertilization occurs, the secondary oocyte becomes an ovum, which will then form the 2-end zygote. So we'll stop here today. Hope you found the lesson interesting, and at your own convenience, do well to look again to all we have lectured, and do well to understand them better. Thank you, and bye bye.